So, real pleasure for me to welcome back Ben Twining to SoMAS. When Ben left here, it was MSRC, it wasn't SoMAS, right? Ben graduated in 2003, was conducting some really state-of-the-art and exciting studies using synchrotron X-ray fluorescence microscopy to kind of figure out the behavior of individual marine protists as opposed to glomming them all together in a big glop and analyzing them as a group. And he made some very interesting discoveries together with Stephen Baines and Chris Jacobson, formerly of the physics department. And Ben has been continuing that work, and it's, I think, pretty interesting, very state-of-the-art type stuff. Ben graduated in 2003, went to Yale for 3 years? 2 years. 2 years. Went to the University of South Carolina for 3 years on the faculty in the chemistry department, I believe, and then has, for the past 4 years, been at the Bigelow lab for ocean sciences in Booth Bay Harbor, Maine, one of the leading marine labs in the world dealing primarily with, not exclusively, but they're well known for their work with marine plankton. And Ben's going to tell us about some of his recent studies all over the world. Ben, thank you.
Thank you. Great. Well, thank you, Nick. Thank you for the invitation. It's nice. Oh, there we go. Nice to be back here. I was thinking about this. I came to walk in my graduation in December, but I think the last time I was really in this building was the day that I was frantically photocopying my dissertation after I'd already defended orally, which was also the day that my first child was born. So I was in the photocopy room, same photocopy room as today. Photocopying, I ran out of the really nice paper that you need, so I went down to Office Max on 347, and while I was walking out with my reams of paper, I got a phone call from my wife. She said, "I'm being induced. Get your ass back here. You're about to have a child." And I frantically drove around because Ferrari wasn't going to work. I lived in New Haven at the time, and I remember. Anyway, so very different day.
Good times.
Good times. Good times. Great. So yeah, so as Nick mentioned, one of the things that I started working on here with Stephen Baines while I was a student was using synchrotron radiation to look at the metal contents of individual phytoplankton and protist cells. And although I actually have done a lot of other things since graduating, I chose to actually present some of our newer results because I wanted to show some growth maybe in the last 9 years. Well, you guys you'll decide if there's been growth. So I've set myself the target of trying to start talking about getting towards the relationship between ecosystem form, and in this case, I'm really thinking about the types of phytoplankton taxa that are present, and the biogeochemical function of an ecosystem. And in this case, I'm really thinking about the accumulation and export and cycling of metals within an a marine pelagic ecosystem. Let's see, I can just do that, right? No, forward. There we go. Okay, so the metals that I'll be talking about today are not mercury, which seems to be what Nick is primarily working on now, but basically first row transition metals that are required as nutrients for a number of processes within phytoplankton. And I've given you sort of a laundry list here. Certainly iron is important for a number of electron transport roles, as is copper. Manganese is required for photosynthesis. Zinc is required for carbonic anhydrase, for alkaline phosphatase, which phytoplankton use to access organic phosphorus. And cobalt, nickel, molybdenum, even vanadium potentially are also required for trace physiological uses within cells. So because of this importance to their physiology, metals can limit the growth of phytoplankton, just like macronutrients like nitrogen and phosphorus can. And this has, of course, gotten a lot of attention, in particular for iron, but there's been some interest and some evidence that zinc may be important in certain places. Gordon and others have been studying B12 and cobalt and looking at that. So there's a general interest in the roles that these metals play biologically, and also increasingly an interest in their geochemical behaviors and cycles as studied through the GEOTRACES program, which I've been part of. I think I also want to really highlight the fact, though, that there are likely many more enzymes and metalloproteins that have yet to be discovered. There was a paper that I really enjoyed that came out in 2010 in Nature by Svetkovic et al. And they took a Pseudomonas, a heterotrophic bacteria, and they did sequential HPLC and ICP-MS. And they tried to characterize all of the metalloproteins that they could detect. And they could only basically identify about half of them. And so their conclusion was that there's a lot more diversity out there in terms of metalloproteins that has yet to be discovered or appreciated. And I think that's a good message to keep in mind as we try to interpret some of these transition metal behaviors that I'll be talking about. Okay, so when I think about biogeochemical function, I'm primarily focused on the role that phytoplankton play as accumulators and then removers of trace metals from the surface ocean into the bottom. So here's a sort of your classic two-layer ocean system. And of course, plankton can have a number of biogeochemical properties, but the one that I'm really thinking of is their sort of role in the biological pump. So accumulating elements, including metals, and sinking and removing them. And through this behavior, controlling the vertical distribution of these metals in the ocean. So obviously, we're very familiar with the Redfield ratio and the relationship between the macronutrients in phytoplankton biomass and their ratios in deep sea seawater. But then I think one of the questions that's really driven me over these past years is, well, what about the trace metals? Do we see these same stoichiometric relationships for these trace metals that are also nutrients for phytoplankton? And what about plankton diversity? I think there's increasingly an appreciation for the diversity in nitrogen and phosphorus ratios that you see in various phytoplankton taxa. And we're also beginning to learn that phytoplankton can also vary in their metal quotas. And of course, this isn't just a green box, but there's a number of, obviously there's a wide diversity of phytoplankton taxa that make up this box of plankton. And Gordon, is this one of your pictures?
No, it's not.
Okay, good. Gordon discovered that the Bigelow annual report contained one of Gordon's pictures last year, and so I'm probably
without
guilty.
crediting it.
Without crediting him, so yeah. Okay, so this idea of an extended Redfield ratio, a Redfield ratio that includes the transition metals, the nutrient metals, was proposed more than 2 decades ago, actually, I believe in 1985. And so let's look at sort of the evidence for that. Here I've chosen three data sets. The first one published, it was a review paper by Bruland et al., which pulled together sort of some classic studies on bulk metal contents in phytoplankton from the Pacific Ocean. Here's a study by Dongyang Ho et al., another Stony Brook alumnus, who grew, I think, 15 different cultured phytoplankton species and looked at their metal quotas. And then a compilation of deep sea or deep Pacific Ocean water dissolved nutrient stoichiometries by Morel. And all of these are normalized to phosphorus in units of millimole per mole, and I'll use that, I believe, throughout the entire talk. So you can see that there are some similarities and some differences. If we compare just the lab and the field studies, so these are the top two, you can see that the copper and the cobalt stoichiometries are pretty similar, both in this field data set and in the lab. So okay, there seems to be some commonality there. But if you look at the iron and the manganese, you can see that they tend to be higher in the cultured phytoplankton, particularly the manganese is about an order of magnitude higher in the cultured phytoplankton on average than in the Pacific plankton. And the zinc and the cadmium are notably lower. And this, I think, has now been recognized to be a function of the choices that these authors or these researchers made in their media. And it highlights the importance of your media selection as you make these measurements, and I think it highlights one of the weaknesses of doing these culture studies. If you're trying to understand the plankton in the ocean, it's hard to replicate the conditions exactly that they're experiencing. If you look at comparisons between the plankton and the deep water, so here we've got our natural plankton assemblage and deep water stoichiometries, again we see some similarities and differences. In the case of zinc and copper and cadmium, you see pretty similar ratios, 3.4 and 3.1, 0.45 and 0.75. And so these are nutrient metals that tend to be very easily remineralized, as I'll talk a little bit more later in my presentation. And they do seem to show very similar behavior in the plankton and in the water. By comparison, iron, manganese, and cobalt are all significantly lower, again, normalized to phosphate, in the deep seawater than they are in the plankton. So you can see that clearly here with iron, 4.1 millimole per mole compared to 0.2. And for these three metals, what's likely affecting this is scavenging, right? So we have scavenging of these metals in deep seawater, which is driving the behavior to go away from this sort of Redfield-like connection. Interestingly enough, if you look at nickel, the nickel is quite a bit higher, at least in this station, in the seawater at depth normalized to phosphorus than in the plankton. And Stephen Baines and I have recently shown the relationship between nickel and silicon in diatoms. And given the location of this is very old deep seawater in the Pacific Ocean. I wonder if there's a sort of connection between the nickel and the silicon there. Okay, so there is evidence for diversity between taxa in metal requirements. And this is a paper that came out right as I was finishing up here by Ho and Quigg et al. Two different papers really talking about the same data. And they made the argument that you can separate, to some extent, the metal quotas or the metal signatures of phytoplankton based on their evolutionary history. So if you separate out the red superfamily and the green superfamily, these are eukaryotic phytoplankton sort of lineages where they had different inheritance of their plastids. And they found that the green superfamily, which includes chlorophytes and presenophytes, or the green algae, they tend to be relatively elevated in copper, zinc, and iron, whereas the red superfamily, which includes diatoms and coccolithophores, tended to be depleted in these. Though I think if you look at the, here I've tried to color code those two groups on the right, you can see even within that sort of generalization, there's a fair amount of diversity between these groups. But quite clearly, just visually looking at, of course, all these bars are not in the same place. There's quite a bit of diversity between these groups that were all grown in the exact same media. So there is evidence that these groups should differ or may differ. Additionally, I think this is a slide actually from my defense. We also know that there is a relationship between the micronutrient requirements and their macronutrient supplies as well. So here are minimum iron quotas, or iron to carbon ratios, for a number of different organisms. And you can see that there are differences for the same organism grown on two different nitrogen sources. So if you look at just a coastal diatom, I think this is a Thalassiosyra model organism, you can see that if you grow them on nitrate, they require more iron than if you grow them on ammonia, because they need iron to synthesize nitrate reductase. Similarly, if you look at the diazotroph Trichodeswww, if you grow it on ammonium, it needs much less iron than if you force it to fix nitrogen to use nitrogenase to do that, which is a very high iron requirement, and they need much more iron to grow. So again, there's an interaction between the macronutrient substrate and the micronutrient requirements as well. And certainly this is true also for zinc and phosphorus, has been shown, and nickel and nitrogen. And finally, there's certainly a dependence of metal quotas on the dissolved metal availability. This is probably the most obvious factor that will affect these quotas. This is a slide that comes from a recent review paper written by Stephen Baines and I. And it summarizes a number of studies, many of them done by Bill Sunda, where they're looking at the behavior of the metal quotas, or the metal stoichiometries. Here you can see for iron, zinc, manganese, copper, cadmium, and cobalt. Again, normalized to phosphorus as a function of the dissolved concentration of either the inorganic iron, what we tend to think of as the free ion, like zinc 2+, or the inorganic species, which are often thought of as the labile species. And in that case, we have Fe prime, which is the sum of the inorganic iron species. And in each of the cases, you can see that as you increase concentration of that presumably available form, the concentrations in the cells go up, right? So that makes sense. The red lines in each of these indicate sort of a one-to-one relationship between that. And you can see that they often go along that line until they get to higher concentrations where the cells tend to regulate those concentrations and they fall off. So we see there's a clear relationship between cellular and dissolved. But this figure also highlights some of the other interactions that are possible. We see, for example, in the case of manganese here, the upper bars, you can tell those are gray. Those are actually grown at a lower light level than the white circles. And so at lower light levels, the same species grown under the same nutrient conditions requires more manganese because it requires more photosystems. In the case of cadmium and cobalt, the cadmium and the cobalt quotas respond very strongly to the zinc concentration because they're taken up through similar transporters. So there's not only relationships between macronutrients and micronutrients, but between various micronutrients and these types of antagonistic relationships. So it's really complicated to predict a priori what your metal quotas in the open ocean will be based on the conditions because of the sort of range of things that can affect it. Okay, so if the goal is to look at the behavior of metals and plankton in the ocean and to try to understand the relationship between ecological community and biogeochemical function, we need to crack open this, calling it the green box now. No more black box. So if we want to crack open this green box and look at the behavior of those various groups, how would we do that? Well, the classic way would be to collect plankton on filters. We all love size fractionated filtration, and you might choose, for example, a very small fraction. If you're a chemist, you might use a 0.45 filter. You might have a 5 micron or something larger, and you'd get sort of 3 operationally defined functional groups of plankton. But we know that these size classes contain a wide variety of metabolic and functional groups within them. So for example, within just the picophytoplankton or the picoplankton, you have heterotrophics, right? We've got our bacteria. You've got autotrophs. Within the microplankton, this sort of middle-sized class, you've got heterotrophs, you've got autotrophs, you've got mixotrophic organisms, and you have autotrophs that may tend to sink more readily because they may be, for example, ballasted, versus ones that do not have a mineral shell and that may not sink as readily. So all of these are being aggregated onto these size fractions, and this really limits our ability to understand the behavior of recycling in the open ocean. So my argument is that if the biogeochemistry is controlled by the taxonomy, then you need to make your biogeochemical measurements on the scale of the taxa. And this is really what motivates us to try to make these measurements on individual cells. One more reason we need this data is in order to ground truth and to assess increasingly complex biogeochemical models that are being produced. So here's an example of a model by Keith Moore et al., where they have, as is pretty common now, they've got a number of different phytoplankton groups in their model. They've got diatoms with a silicon requirement, diazotrophs with a high iron requirement. They're small phytoplankton. They've got a number of different zooplankton. And of course, each of these is parameterized individually. They're using different metal quotas and requirements. And so we want to understand, do the variations that they predict actually play out in nature? And then also, can we help them better parameterize these models by actually measuring the quotas in the open ocean so that we know what the cells are actually able to obtain? Similarly, I'd say there's a new trend towards also sort of trying to go back to almost first principles when modeling phytoplankton and look at the various, in some cases, sort of biogeochemical processes that are going on in those cells and try to understand the impact of each of those on metal or macronutrient biogeochemistry. And here again, we want to understand how the metals are behaving in the cells that may have different types of activities going on in them. So for all of these needs, we're trying to produce more informative understanding of the metals in cells. Okay, so how do we do it? Well, the approach we've taken, as Nick mentioned, is to use synchrotron radiation to measure the elements in individual cells. It's actually fairly straightforward. We collect our samples with clean technique. This is fairly standard these days. We immediately centrifuge them onto electron microscopy grids for the most part. In some of the cases, we actually place them individually for Trichodeswww. But we put them onto EM grids, which are coated with a carbon film. And then we image them, usually at sea, while their chlorophyll fluorescence, for example, is still present. And we can pick out the samples and the target cells that we're interested in. And then we store them until we put them in front of a highly focused monochromatic X-ray beam. So this is produced at a synchrotron, which I'll show you in a second. And basically, we scan an X-ray beam over individual cells to produce fluorescence. The instrument that we've used almost entirely is the Advanced Photon Source. It's at Argonne National Lab outside of Chicago. It's a 1.2 kilometer ring, and I've put the Roger Revelle in the middle as a scale bar here. And it's basically a ring around which you accelerate electrons. And the purpose is to bend the path of those electrons with magnets and therefore generate high energy and high intensity X-rays that can be used for a suite of analytical uses. Most of the work we've done has been in Sector 2, shown here, and this is an example of the experimental hutch. So this is a lead-lined room within which we place the sample, and then the doors close, and there's lots of foreboding alarms that go off to make sure you're not inside. And then they basically allow beam to come into the room and to irradiate the sample. And this is an example of the beam. It's all under high vacuum coming into the hutch. And here's another schematic. Basically, you have the beam coming in. The energy is tuned using an undulator, which causes the electrons to wiggle back and forth. And then it is focused with a Fresnel zone plate. And there's your sample right there. And we measured both the fluorescence produced by the atoms in the cells, and we also actually measure the X-rays that are transmitted through the cell in order to get a sense of the biomass of the cell. So we're taking both transmitted and fluoresced X-rays. And so using this, we get a two-dimensional map of the elements in the cells, and in some cases three-dimensional now as we are starting to embark on tomography. And here are some classic pictures. These are definitely from my dissertation defense of those elements in the cell. So we get both qualitative information of maps. So here's a silica flagellate from the Southern Ocean. It's about a 20-micron cell, and you can see it has an opal frustule or shell. It has chloroplasts centered in the middle, and you can see the distribution of some of the bioactive metals map onto those features in ways that make sense. For example, the silicon is on the shell. Sulfur is largely located in the middle in the cytoplasm, largely probably in the protein fraction, and iron and zinc in this case are largely localized in the presence or the locations of the chloroplasts. We also get quantitative information on sort of the suite of elements between silicon and zinc. These are at least the easiest ones for us to get using the typical setup. And so these are the fluorescence peaks averaged or summed over the entire cell that we can then fit with peaks and quantitate for each individual cell. I should point out, this technique does not give us carbon or nitrogen. So two of maybe the most important elements in terms of cell biomass, unfortunately we don't get them. We get phosphorus, we get sulfur, which we often use as a biomass proxy as well. But carbon and nitrogen fluoresce with such low intensities that they're difficult to measure with this technique. Okay, so time for an outline. Here we are, 20 minutes in. So I'm now going to nix that an hour and a half. So I assume that's, everyone's eaten already, right? Perfect, great. So we'll see how many of these I get through. I've got sort of 4 vignettes of examples of data collected using this technique, building sort of over time as we've collected data from different ocean basins. First, we'll be looking at trying to pull together data from, I guess, 4 or 5 ocean regions to look for both taxonomic differences and differences across ocean basins. And then, in the section of the talk where I present the data that I've worked up on the plane on the way here, I will be showing some data that we've just been getting from the recent North Atlantic US GEOTRACES cruise, where we have cell quotas and also bulk data. And then I want to talk about the fate of metals in sinking diatoms and some work we did on the spring bloom a couple years back. And if I have time, we'll talk about Trichodeswww, but I have a feeling that might fall by the wayside. Okay, so here is a map of the locations of the studies that I'm going to show you data from in the coming slides. So these are locations where we've collected cells and analyzed them with X-ray fluorescence, synchrotron X-ray fluorescence, or XRF. This includes the SOFX experiment down here in the Southern Ocean. This is work that I performed well as a student here, both north and south of the polar front in these high nitrate HNLC waters. Data collected during the FeCycle II project off the east coast of New Zealand in 2008. Data from the equatorial Pacific in a project with Stephen Baines and Nick Fisher here, looking at metal cycling and silica limitation in the equatorial Pacific. And then a couple of studies done in the North Atlantic, 2 of them in the Sargasso Sea at the BAT station, where we have focused either on Trichodeswww or on Synchrotron. And then, most recently, the US North Atlantic GEOTRACES program, which actually spanned across the North Atlantic. So I'm going to pull these together as examples of these ocean basins to try to look for some patterns and differences in the metal quotas. So starting with iron, I'm going to show one of these figures for each of the transition metals. So I think they all have the same formatting. Here we've got iron expressed as millimole per mole phosphorus for 4 different taxonomic groups: diatoms in black, autotrophic flagellates in red, autotrophic picoplankton, primarily Synchrotron in green, and just in the North Atlantic at the BAT site we have Trichodeswww data in yellow. And on each of these I've included a dashed line to indicate the sort of extended Redfield ratio as proposed by Bruland et al. in that early bulk paper. And I'll try to sort of summarize some of the trends as I see when I squint my eyes. And if you're looking for a more sophisticated statistical analysis of these, I will merely state that I was trained in a laboratory that has on the door of the lab a bumper sticker that says, "We don't need no stinking statistics," and refer any further questions to Nick on that. Okay, so what do we see? Well, on the top I've got some taxonomic differences. On the bottom we've got the geographic. And clearly Trichodeswww here has a very high iron quota. This is not a surprise to anyone who's studied them. They have, again, a very high requirement for nitrogenase when they're fixing nitrogen, and they have a lot of iron. Next, we tend to see elevated concentrations in diatoms, at least in 2 of the 4, we'll call that half, studies where we see elevated quotas in diatoms relative to the other eukaryotic phytoplankton. We see that in the equatorial Pacific and also in the North Atlantic. We tend to see the lowest in autotrophic flagellates, and at least in 2 of these 3 studies where we actually have cyanobacteria, they also seem to be somewhat elevated. Though again, the differences are more subtle and may not be statistically significant. I've just added this North Atlantic data, so we will get to the statistics. I'm not trying to be flippant about that. But also, if you compare within taxonomic groups, it does appear that the North Atlantic may at least certainly it shows elevated concentrations in the diatoms relative to the other regions, followed by the Pacific Ocean and then the Southern Ocean, which is a well-known HNLC region. We do see the lowest iron quotas. So there does appear to be an oceanographic gradient going from the high iron Atlantic to the low iron Southern Ocean, which matches our expectations based on sort of the understanding we have of the metal delivery to these basins. But it's really, I think, cool to show that there's actually, we're seeing a signature that in the biota, that the biota are responding in a sort of a community fashion. And just for interest, let's go back and compare this to what Ho et al. Presented. They actually predicted, or they found the lowest iron concentrations in the diatoms compared to the other groups, whereas we're actually seeing the opposite, at least in several of these studies. And I'll leave it at that for now. We're happy to talk about that later. Here's zinc. Zinc, again, the data span the proposed extended Redfield ratio. But again, we see some significant taxonomic differences, in particular, that the diatoms, and to the extent we have data for Trichodeswww, appear to have more zinc than the other cell types. And the I would say there's not much of a difference. We don't really have a lot of data to look at differences between autotrophic flagellates and cyanobacteria. But we also see, I'd say, a pretty significant difference between groups in that the Southern Ocean, these big groups here, the diatoms in the Southern Ocean tend to have more zinc than the diatoms in the other regions. And we'll see this borne out in the bulk data as well. Again, they predicted low zinc in diatoms, which conflicts or contradicts our data set here. Here's manganese. Manganese, the taxonomic differences tend to vary between regions, so I think it's more difficult to draw conclusions there. We do tend to see elevated manganese in the equatorial Pacific. I don't have a great explanation for that, and in fact, I'm surprised we didn't see it in the North Atlantic, because I'll show you in a little bit. We saw a lot of manganese in the North Atlantic. But in general, there's probably fewer gradients there. And here's cobalt, which we have less data for. Cobalt not only occurs at about an order of magnitude lower concentration in cells compared to those other metals, but it also is more difficult to analyze because of some spectroscopic details with the X-ray analysis. So we have less data there. But the interesting thing that comes out is that if there is an effect, it tends to be that the cobalt is enriched in the flagellated cells. So the other metals we saw generally were enriched in diatoms. And here you see a different trend. And given the interest in B12 limitation and cobalt cycling, it's just interesting that there's a notably different taxonomic signature there. And interestingly, the Ho et al. Their data set did show, at least in the dinoflagellates and the coccolithophores and heptaphytes, elevated cobalt compared to the diatoms. So maybe there's an agreement there. Oh, and finally, nickel. Nickel, again, we see a span of the proposed ratio. We see lots of nickel in Trichodeswww. I think this is primarily, well, it could be due to either urease, which is a nickel enzyme, nickel form of superoxide dismutase, or potentially also nickel in a hydrogenase. So hydrogen is produced as a function of nitrogen fixation, and it may be that they're dealing with that hydrogen there with the nickel hydrogenase. But we also see elevated nickel in the diatoms. And at least in the Atlantic, we see quite high nickel in the cyanobacteria as well, which is predicted based on their nickel superoxide dismutase presence as well. So, I got a little before we get to our conclusions here. Now let's compare this to what we know from published bulk data. So I've shown you some ocean basin differences as a function, or as shown in the individual cell data. But we can also look for differences in p-normalized metal stoichiometries from dissolved nutrients. So here we're getting at that other side of the sort of the Redfield effect, where we're looking in the ratios in the dissolved in the water column and looking to see if we see differences between basins here. So here I've got, this is from the review paper that's just come out by Stephen and I. Top row is North Atlantic Ocean data sets that we've summarized, Pacific in the second row, and Southern Ocean. And here you can see elevated iron in the North Atlantic, depleted in the Southern Ocean, matching the trend. We see, although there's certainly some variance in these results, we do see the highest zinc to Fe ratios in the Southern Ocean compared to the other basins, and less, I'd say, weaker gradients in the other elements. The cadmium, which we can't detect with the synchrotron, is also elevated in the Southern Ocean. But so there's some evidence here from the flip side that there are real differences in how the metals are cycling in these ocean basins. It's not just the cells. We're seeing the difference in the biogeochemistry in depth, too. Oh, there we go. How do I do this? OK, so sort of conclusion of this first part. We do see group differences in the ocean phytoplankton that differ from the lab studies. In particular, we see many of these iron, nickel, and zinc elevated in diatoms compared to the other cell types, and in some cases in Trichodeswww as well. So there does seem to be a real difference between the way this ballasted, potentially biogeochemically unique group of diatoms is behaving compared to the other cells. And it differs with cobalt. We see the opposite effect. And additionally, when we look both at the synchrotron data, at dissolved metal profiles, and we also can see this in the bulk particulate data, which I took out for the sake of time, but all of these show an interocean gradient in metal behavior, particularly with iron elevated in the North Atlantic and zinc in the Southern Ocean. So I think we're seeing a really nice connection between the biology and the geochemistry here. OK, now I'm going to show some data just from the North Atlantic. And the goal here, from my perspective, was to look for a gradient across an ocean basin. So since we started this work, one of the things we've been looking for is differences in the ocean as a function of the environmental parameters. And so I've just talked about very broad scale differences. And now we're going to look for a difference that goes just across the Atlantic. So this is a map of the US, not just the US, this is the International GEOTRACES cruise plan. GEOTRACES is an international program started maybe a decade ago with the goal of constraining trace element and isotope fluxes and cycling within the global ocean. So pretty high goals. And all of the black lines here show completed cruises, actually black and yellow, and the red are all of the planned cruises yet to come. So you can see there's an enormous effort going into it, and the first US effort was the cruise going across the Atlantic Ocean, which our group participated in. So here is a cruise map. I was unable to find the ODV file for this, so this is a scan of our original figure that we were working on, and I've even, you can't quite see the coffee stain, but there's a nice coffee stain there. So this cruise ended up being 2 cruises because the ship broke at the middle of the first cruise. So originally, in the fall of 2010, we left from Lisbon, steamed out into the North Atlantic, down to the Cape, actually in towards the African upwelling zone, and then out towards Cape Verde. At that point, the right thruster on the RV Nor died, and the ship limped back to Charleston via this route. And actually, one of our group members stayed on, and we collected some samples from the surface water during that leg. A year later, the rest of the cruise was completed, starting at Woods Hole, going down, this is line W, going down to the Bat Station, and then going across toward Cape Verde Islands. And I've pointed out in yellow here sort of the touch points for stations that I will highlight in the data with what in Woods Hole, the Bermuda Atlantic Time Series, the TAG Hydrothermal Vent Station, the Cape Verde Islands, and Lisbon. And at each of these stations that have a triangle around them, we collected samples for synchrotron analysis, as well as for bulk particulate analysis, both from the surface mixed layer and from the deep chlorophyll max. And for the sake of this, I've just taken the mean of those. Each of those filters was digested in 2 ways. First, we used a hot acetic acid and a hydroxylamine leach, which is intended to get just the biogenic fraction, sort of an operationally defined biogenic fraction. And then the filters were digested with a very strong multi-acid leach to get the rest of the stuff. OK, so here, I'm going to show this also. I'm going to go move to this sort of formatting of the slides and show this for a number of different metals. This is for iron. And on the left, I've got the iron to P ratios for 3 different fractions. The red is the bulk particulate material as obtained with our labile leach. The black is the total, so I've taken the sum of the labile and the total leach, and the blue is the synchrotron data, all, again, presented as millimole per mole phosphorus. On the right, I'm presenting just the synchrotron data, and in this case, I've normalized them to sulfur as opposed to phosphorus. And I do this because sulfur provides us with an alternative biomass proxy that tends to be less noisy, and it doesn't respond to gradients in dissolved phosphorus availability. So, of course, phosphorus is another nutrient which has its own gradients, and the cell phosphorus quotas respond to that, but the sulfur quotas probably should not, or we would expect them to. And so it's just an alternative way of looking at it, which provides a little less noisy data set. No intended bait and switch here. Okay, so what do we see? Well, looking at the bulk data, we see a couple things. First of all, the black dots are, for the most case, significantly elevated over the red ones. So most of the particulate iron present in the Atlantic Ocean is in the form of a lithogenic form. It is not associated with the biogenic material. And we can see that also if we plot iron as a function of aluminum in the particles, they match up right along their crustal ratio. So most of the iron out there is not in cells. It is in lithogenic material. Additionally, if we look at the comparison between the blue and the red, they're within the same ballpark. So this labile fraction that we've gotten with this weak acid digest, gives us a pretty good approximation of what we see with the synchrotron, and also here's again this extended Redfield ratio as proposed by Bluland. What I think is most exciting is looking at the zonal gradient that we see in the iron quotas. So starting in the west side of the basin at Woods Hole, and moving towards the east, we see quite a notable decrease in the iron quotas of the cells. And here I've grouped all of the cells together. I haven't had the time yet to pull out the different taxonomic behavior, so I'm not sure what contributes to that. But most of these cells are autotrophic flagellants, and they're clearly showing a gradient across the Atlantic Ocean, which is really interesting, I think. All right, here's data for manganese. Again, another nutrient element needed for photosynthesis. And here, we see that most of the particulate data, the red and the black, most of the particulate material comes out with the labile digest. So this, again, isn't surprising. Most of the phosphorus should come out with the digest, but also manganese and even manganese oxides that are produced autogenically in the ocean should be acetic acid labile. So that's not surprising, but it's nice when things match your expectations. And we see a very strong west to east gradient in bulk manganese to P ratios, all of which, interestingly, are very significantly elevated above what we see in the plankton. Now, if you look just at the plankton, we again see, although a little bit noisier than the iron gradient, but we see again a gradient from west to east in the cell quotas. So we've got a gradient in the bulk particles, most of which are not cellular, and in the cellular fraction as well. So these are, I think, very exciting. We often haven't seen strong gradients within a single ocean basin, and I think this is the first evidence of that, which at least gets me excited. All right, here's cobalt, and cobalt shows something different. Here we see the bulk data matching pretty well with the single cell data, so there doesn't seem to be a lot of abiotic cobalt out in the ocean. And we see, again, if you squint your eyes and sort of tilt your head back, sort of a bowing out with the highest cobalt to P ratios in the middle of the Atlantic Ocean basin. And you'd have to squint your eyes even more, but there's the hint that may be seen in the biogenic data as well. So it's interesting. So there's clearly a different source for cobalt compared to iron and manganese, or at least it's maybe different sinks for this element, but different than manganese and iron, which is really interesting. And you do tend to see it. If anything, you may see it in the cells as well. And that's different from the nickel. Nickel, we see less agreement between the bulk data and the single cell data. And then they also show this different behavior with the nickel data bowing out in the middle of the ocean, in the middle of the North Atlantic, and the cells showing sort of a dip, and you can see that here in the sulfur normalized. So we've got some elements where the bulk data tends to really track the cellular data, and ones where it doesn't. And I don't have great explanations for this yet, but I think there's clearly very different metal cycling going on between these different metals, and I think there's hopefully some interesting things to be learned about the role of the plankton in that. Oh, and zinc. Zinc is by far analytically the biggest challenge to study in terms of both its very low levels in surface waters and its proclivity for contamination. So it's a much noisier data set, but we do tend to see pretty good agreement between the bulk data and the cellular data. And I don't know if that's real or not, but there clearly is not the same strong west to east gradient that we see with iron and nickel, excuse me, and manganese. Okay, so what can we sort of preliminarily conclude from here? Well, I think there are some real zonal gradients in metal quotas just across the North Atlantic Ocean. So first we talked about North Atlantic versus Southern Ocean. Even with the Atlantic, we see these differences, particularly with iron and manganese elevated to the west, cobalt elevated in the middle, and nickel showing sort of two behaviors, whether you look at the bulk particles versus the cells.
Ben, can I ask a question?
Please do.
Is it really that the nickel is elevated or just the phosphorus is depressed? Because you're doing ratios, not
right. So I admit that that's a very good question. I don't know the answer to that, but if the phosphorus was driving that, I might expect to see that in some of the other elements as well. So I mean, it may not. The signals, cobalt is a very simple one to measure actually, and so yeah. So I mean, could this be driven by heat?
The might mask that effect because there's more of the other elements or something.
Certainly, you would expect biomass to be highest at the margins. And it's certainly something we need to look for. You're right. No, but sorry, go back here. I think I would have expected to see it in some of those other ones as well. But you're right. It could. There certainly could be. It would not explain these, which are of course driven by or within each single cell. And I think this is maybe a little more technical, but it's also useful to know that this digest, which was developed by a student of Ken Brulin's, published a couple years ago, does seem, except for manganese, to give you a pretty good approximation of the metal quotas. And that's been important because we are one of the only, if not the only groups looking with this single cell technique. And it's important that others that we be able to broaden the coverage of data beyond just what we can do with the single cell technique. And that's showing pretty good promise, I think, for that. Okay. So I want to move now. So we've sort of tried to give two examples of ways where we can look at individual cells and start to examine how metals are accumulated within phytoplankton in the natural environment, particularly across these larger gradients. I want to now move to a different example, and this is a study where we're then trying to look at the other biogeochemical function here, which is the removal of elements and metals as cells sink out from the surface water. And in this case, we were able to collect samples from a spring bloom off the coast of New Zealand and look at the fate of the metals in just the single diatom taxa as it sank. So this was performed as part of the FeCycle II project, which was led by Phil Boyd down in New Zealand. And it occurred in the fall of 2008. And so here's your map of New Zealand, South Island, North Island. And this is the region of the study. It was 300 kilometers to the east of New Zealand. And we first tracked the water mass and were able to find an eddy within an anticyclonic eddy. So that allowed us to have a situation where we were trying to constrain the mixing of this water mass with the water masses around it. So we could follow a single water mass without adding a tracer for the period of the bloom. And you can see, excuse me, the black here are the currents, vectors for the currents, and the blue is the ship's track over the course. And you can see that there was a strong wind event which blew, actually this is the ship, but it was following a drifter. And this drifter gets blown out of the eddy at one point, which I'll point out here. So here is this is density sigma theta as a function of time. So these are all in year day or Julian days. And you can see that there's a period at which you have stratification of the water column right here. So we were present, and then suddenly we see stratification, and this is really when this diatom bloom occurred. So you can see that in the density, and you can also see it in the transmissometry. So this is showing us the presence of particles in the water column, and you can see that that corresponds very closely with the onset of stratification. So we were able to catch a bloom of Astronella, actually it's Astronellopsis, excuse me for that, it's been renamed, glacialis, which is a large triangular diatom. You can see a picture of it here. And this is a map of the cell abundance. This is from microscopic cell counts as a function of date. And well, I'm not sure which is which, blue, white or black, but here it is in cells per mil. You can see that there was sort of a pre-bloom, and then it bloomed again, and then we saw this drop. And here it is as a percentage of the microplankton community, and you can see that it peaked out at about 65%. So it was a significant part of the plankton community. Here is the greater than 20 micron chlorophyll, and so here you can see those features again. And these are the periods during which we took our samples. So we collected samples from the surface mixed layer and using GoFlow bottles, and then we also deployed trace metal clean sediment traps that were floating at 100 and 200 meters. And those traps were out for 3 days, so from the 267 to 270, and then they were recovered on day 270 right at the bottom of this balloon. And what we were able to do is actually observe these diatoms at each of the depths, in the surface mixed layer actively growing, or at least present, and then in the traps at 100 and 200 meters. And because they're easily identified from their morphology, we can look at the elemental composition of just the cell without having to normalize to biomass. And so that's what we did. We also collected samples for ICP-MS. And here is what we see. So here I've represented phosphorus, sulfur, silicon, iron, zinc, and nickel. These are quotas in femtomole per cell as a function of depth. And on the bottom axis, I show the fluxes of these calculated from ICP-MS bulk data, and these are shown in white circles. And you can see that there's rapid remineralization for phosphorus, sulfur, nickel, and zinc. And I should say, we see the sulfur remineralization or loss from the cells in the individual cells, and we see the nitrogen loss in the bulk data. But clearly these are lost quite readily. You see about 75% of cellular phosphorus lost between the cells at the surface and the cells at depth. And you see quite different behavior for the iron and the silicon quotas. So here's silicon. You can see that at least within the cells, again, it sort of bumps around, but they generally are retaining their silicon, which is really interesting because when you compare it to the bulk silicon data, you can see that there's a strong loss of bulk biogenic silicon. So silica is being remineralized, but it doesn't seem to be remineralized from these cells that make up the dominant part of the bloom. Here's the iron quotas. You can see they also stay relatively consistent, both the black data showing the cell behavior and the white data showing the bulk fluxes. So iron is not being remineralized, at least in this upper 200 meters. So another way we can look at this is to calculate and look at the stoichiometry of the cells and compare it to the stoichiometry of the material that's being lost from the cells. So that's what I've done here. On the left, I'm showing iron normalized to phosphorus, both in the cells at the surface in the green bars, in the 100-meter cells in red, and then sort of the difference as shown in black. So we've calculated this both by looking at the loss of iron and the loss of P, and then represented that as a ratio. And you can see that the remineralized material looks very different from the cellular material in the case of iron, whereas in the case of zinc, they're relatively close. Basically, the zinc to P ratio of the material matches what's in the cell. Now, whether that demonstrates that they're being lost together, obviously you can't prove from this, but clearly they're behaving very differently from iron. We see this also in the bulk data down here. What's interesting, if you look at these are iron to P for the bulk particles, you can see that it is going up rapidly. So clearly, the phosphorus is being lost and iron is being retained. And also you're getting scavenging of additional iron fractions in the bulk data. Yes?
I just have a quick question. When you say loss, isn't it like a net change because you could have precipitation and things like that on the bulk particles?
Yes.
You're really looking at net change.
Thank you, yes. And I'll have to get back to that. Because yes, so we're looking at net change because if materials were, for example, scavenged back onto cells, we would see that here. And that's exactly what we think is happening in the iron, in fact. Which I'll get to in 1 second. So the other thing we've tried to do is try to use some of the spatial data that we get with this mapping approach. And so what we've done is looked at the correlation between the metal distributions, for example, in this cell collected from 30 meters, and the concentrations either of the metal on a per pixel basis, and either our biomass proxy is phosphorus or sulfur, or silicon as a proxy for the frustral of the diatom. And in surface cells like this one, you can see, for example, iron and sulfur are quite closely related within the cell. So here, each of these data points is a pixel that we've extracted from this map. And you can see that areas, that basically they map fairly well onto each other. If you then compare that to a cell collected from the sediment trap, for example, this one at 200 meters, you can see that the relationship is broken down. And there's areas that have very high sulfur, but not high iron, and vice versa. And so our sort of goal here is to try to use this to squeeze out a little more information about what's going on with these metals as the cells, and these elements as the cells sink. So this is an attempt to represent that in a graphical form, which is maybe not so digestible in the next 5 minutes. But we see a couple of trends. First of all, what we've shown here is 3 different metals normalized either to phosphorus in the top panel, to sulfur in the middle panel, or silicon in the bottom panel. And then 3 different depths, surface, 100 and 200 meters. And a couple things pop out at me. First of all, is that and these are the coefficients of determination, the R squared, for each of those linear regressions, and then the means and the standard error for multiple cells. And you can see that these relationships decline with depth, both for metal versus phosphorus and for metal versus sulfur, and to some extent for metal versus silicon as well. So as the cells are being degraded as they sink, basically the elements are becoming less closely correlated spatially in the cells. Additionally, we see a couple that tend to become spatially decoupled, and that is, that is in particular iron and phosphorus. So you can see here's iron. Actually, it's not difficult to see that from there. But also, as the cells sink, the iron tends to actually not become less spatially correlated with the silicon. Okay, so we're seeing the drop in the association of iron and zinc with the biomass metals, but we continue to see a relationship, basically an equally strong relationship between iron and silicon. Okay, so what we think may be happening here, here's my arm waving portion of the talk, that we have this potential remineralization scenario where the phosphorus and nickel and potentially also the sulfur may be lost from the membranes. So this loss is occurring relatively rapidly. And additionally, that the zinc and the phosphorus and the sulfur are likely to be lost from nuclear material and proteins. And that this may explain the very close relationship spatially we see between zinc and phosphorus even as the cells sink. For the iron, as Cindy pointed out, we can't separate loss from the cell versus scavenging, or iron that is lost and re-scavenged. But certainly the retention of a spatial relationship between iron and silicon could be explained by iron that is lost being scavenged back onto the frustral. Additionally, it's been certainly well known for a number of years that the loss of silicon from sinking diatoms is controlled partially by the degradation of the organic coating. So that probably explains the slow loss of silicon in the cells. But the end result of this is the preferential export of iron relative to carbon and the other major nutrients. And this has been suggested at, and there are some data sets of dissolved iron distributions which indicate this, but I think this is the first time where we've looked at it from the particle side and actually tried to see the difference in the behavior of iron and the major nutrients in sinking cells. I think that's an advance we've been able to make using the single cell technique. Okay, in the interest of everyone's tummy, I'm going to skip the last one, Trichodesmium metal quotas, but I'll give you the take-home message, which is that the trichodesmium contain more vanadium than iron. So how's that for exciting?
More vanadium?
More vanadium and iron. Yeah, so we've done a number of work with both synchrotron and ICP-MS data, and we've seen, as you can see here, in each case, in almost all these cases, actually more vanadium in the trichode than iron. This is work done by my postdoc, Jochen Neuster, and a paper about this came out in Frontiers this year. So we'll skip this, but there's other cool things we're seeing in terms of some different elements and some unexpected relationships.
What's the dissolved concentration ratio of vanadium to iron?
Well, so here's dissolved vanadium. Good question, Gordon. So here's dissolved vanadium. It's almost conservative in the water column. It's mostly in the form of an anion, it's vanadate. But in some cases, you do see a little bit maybe of nutrient uptake at the surface. And the explanation for this, there's a couple potential reasons. One is that the trichodesmium may be using a vanadium form of nitrogenase. The genomic information, this is work done with Adam Kuska, another Stony Brook alum. And his work on that suggests that that's not what's going on. We don't think that trichodesmium have a vanadium nitrogenase, but there is a form of nitrogenase that uses vanadium. I think the most likely is that the cells are using it as a vanadium haloperoxidase to counteract oxidative stress. in the surface water, so that's potential. But it's also potential that they're taking it up accidentally with vanadate as an analog for phosphate because of very low concentrations. So I'm sorry to rush through that, but let me go to my overall conclusions to finish up here. I think in summary, phytoplankton metal quotas can vary significantly between phytoplankton groups in the ocean, and certainly in my my first sort of vignette was focused on that, but even the part about the Trichodeswww I think also demonstrates that these groups can have very unique metal signatures. And the consequence of that is that community composition can affect metal cycling. And I think that's an important conclusion. Metal plankton quotas can also vary significantly between basins and along gradients within an ocean basin. And this will also, I think, hopefully as our understanding of the metal cycling becomes more mature, we'll understand what drives those variations. And finally, vertical fluxes may also, in fact, are likely to be influenced by community structure. Often we think that ballasted organisms may play a unique role in flux. And so therefore, we're starting to look at the relationship of these metals and these macronutrients with sinking material to try to understand their behavior in those. And with that, let me give a very hearty recognition and thank you to my collaborators, in particular Stefan Vogt at the Advanced Photons Force, Stephen Baines of Stony Brook, who have been partners in this work throughout it, lots of folks who have helped with field sampling, my postdoc Johan and Adam, who have worked on the Trichodeswww with me, and Andrew King, Scott Nodder, and Phil Wood should be listed there for the FeCycle II. And I'd be happy to take any questions. Thank you. If you all can stand it. Yes?
Okay, so clearly you have to normalize to some element to get these quotas. But phosphorus, are you going to abandon that? Because that's troublesome.
Because it's so variable?
So variable, and it's limiting in many environments, and cell quotas within a species vary, and within different clades they have different phosphorus quotas. Sulfur seems like a better bet.
So
it's never in limiting concentration.
Right.
All you need is the energy to reduce it down to -2
right.
oxidation state.
So I really like sulfur as a proxy for those reasons, and we haven't gotten too much pushback from reviewers for doing that. We've made a number of arguments for why that's a good relationship. There is some culture data supporting the pretty close relationship between phosphorus and sulfur in replete cultured cells. The issue is always DMSP and DMS in cells. Certainly if you get a high DMSP producing phytoplankton, that could be a factor. My impression is that, so all of these are fixed with glutaraldehyde, and my impression is that we actually probably lose the DMSP during the fixation because we haven't seen strong evidence of that.
That'd be a good thing to check.
It would be a good thing to check. So yeah, but that's the risk there. That will become more important. I've gotten samples where we're really focused on the coccolithophore fraction, and those we need to be more careful about that. So we will do that checking. Yeah.
But just on that point, isn't the sulfur primarily associated with amino acids and proteins in the cell? And if there's not enough nitrogen, then the cells can't make, which occurs, right? There's not enough nitrogen, the cells can't make protein, and so they'll limit the amount of sulfur they take up. And so, as a consequence, sulfur may be as subject to variations in nitrogen as phosphorus is also varying. So, because there's not a lot of sulfolipids in marine organisms, so sulfur is virtually all in amino acids.
Yeah, though I've seen some of the recent sulfolipid work. I think.
But well, there's some.
Yeah, there's some.
But the proportion of sulfur associated with sulfolipid is very low compared to protein.
Certainly that's an argument that sulfur and nitrogen should track each other pretty well. So I guess it's possible if you have nitrogen effects on protein content that you would see that in the sulfur. Yeah, we certainly have to think of that.
Think about it.
Think about it.
You got one?
Yeah, you showed that nice trend of the iron
Right down in front. Excuse me, yes.
You showed the nice trend of the iron quotas going across the Atlantic. When was that conducted, and do you think there could be any seasonal effects?
So good question. So there were two cruises which I've combined there. They were both done in the late fall, not a great time for phytoplankton research. So late October into November. So I would expect there to be seasonal effects. Certainly the iron dust deposition to the North Atlantic will have seasonal effects. I don't think that the fall is a particularly strong time for dust. I think most of the dust is deposited at least around the Sargasso comes in the summer. So I don't know what the residence time of that would be, whether you're still seeing vestiges of that. But certainly I think you would expect to see, I think you'd also expect to see some meridional gradients, as you cross the sort of high dust region. And I've completely glossed over that by plotting them simply as a function of distance along the track. So we need to refine that a little better. And look across, for example, as you go from Lisbon down to basically going due south. So I think there's probably more refined spatial analyses, but also temporal that we could do. Yeah, but most of the dust is coming from east to west. We've got this sort of high gradient in the west. I don't know if that's what you're seeing as shelf derived. I don't know. It'll be interesting. So the goal of the GEOTRACES program is to bring together dissolved metal and particular metal analyses for a whole suite of elements. And so all of those are due on December 31st this year. And it'll be really interesting to tie it together with the dissolved, also for cobalt nickel, see how the distributions change. There's other hands here first. Yes, Cindy.
Since people had problems with normalizing the phosphorus and sulfur, I thought I would point out a problem with silicon, normalizing the silicon. And that's a recent graduate of ours, Planktonic, has found that magnesium and silica accumulate on the degrading cyanobacteria and increase with that. And so that also might explain why why your silicon doesn't go down as fast. It may not actually be dissolving more slowly, although it probably is, but it may also be accumulating as well. And that's...
Yeah. So you're talking about the lack of a drop in silicon. So that's just in the diatoms we're looking at it. So it would have to be, it would have to be silicon precipitating through those same mechanisms onto the diatoms, which could be happening. But I'll bet that that's a small factor of all the silicon associated with the diatom.
Cyanobacteria is quite large, but I have no idea about diatoms.
Yeah, so that was just specific to that diatom.
They do have an extracellular polymer coating, which is what first
Mm-hmm.
It'd be interesting to find out.
Yeah, no, I think there's all sorts of interesting things with silicon that, yeah. Yes.
You mentioned iron scavenging. What is scavenging? Is it a physical, a biological process, a
That's
chemical
a
process?
good question, and everybody uses it differently. Yeah, sorry. So when I think of iron scavenging, I'm referring to an abiotic adsorption onto particulate materials. So some people will use it to include biological processes that take an element from the dissolved to the particular phase. I tend in my brain to separate those out. So what I'm thinking of when I refer to that would be, for example, iron that is, in the case of the sinking project, iron that is being remineralized through bacterial activity from the sinking diatoms, and then once it's in the dissolved form, is then sticking back onto the outside of the cell.
So it's now Fe3 something.
It's Fe3, and it's not ligand bound, or those ligands are degraded by the bacteria or something, and it's sticking back to the cell. And I think also, talking about the deep iron to P ratios where I mentioned scavenging is probably removing iron. I think that's also what I'm thinking of is, we know that iron, looking at the relative ratios of iron and iron binding ligands, the excess ligands tend to go away at depth, and it's much more likely that iron is scavenged abiotically at depth.
Do you see localizations of iron that might be consistent with chemical precipitation?
So the sediment trap diatom samples there is certainly a much, I'll say, messier distribution of, for example, iron in the cells than at the surface. And in the surface, they tend to map onto sort of biological, looking at the chlorophyll, for example. There are definitely features, iron-rich features, which do not seem to be associated with visible cellular features. So I think whether that is, it is hard, these have been sitting in a trap only for 3 days, they're unfixed. But yes, I think the answer is that there is evidence for adsorbed iron or externally associated iron that doesn't map intracellular patterns. Yeah, but those are tricky to pull out, particularly in 2 dimensions. I mean, I try to be a little soft pedal that a little bit. But you do see an increased occurrence of that kind of a feature. Yeah.
Could the contribution, the volume of hydrothermal fluids and the weathering of relatively young ultramafic basalt be a factor in the nickel data you get?
Right.
It's high in the middle of the ocean, which seems to be completely decoupled from
from the other elements. So my understanding is that most of the geochemical signal, those hydrothermal regions, does not make it up. I mean, there's a huge distance and a lot of density gradient that are precluding that from mixing into the surface layer. So I think that would be a very novel finding if you were able to show that. I know that, for example, people have looked at iron isotopes where you see a strong iron isotope concentration. It moves laterally along isopicals, and you see it over, for example, like that. So I doubt it, but I'm going to say no. Yeah.
Okay. So you compared some of your ratios to the Quig and the Ho papers. Is that a valid comparison? I mean, to my way of thinking, your single cell analysis really is a snapshot of telling us what the cells are exposed to and how they respond. Whereas my recollection of those papers is that they were culture work, and they're subject to whatever the media is.
Of course.
Did they use Sunda's approach? Or was it
So for the Quig and Ho data set, they used a single, one concentration, one media. They used it for all of them, and they tried to match it to their sort of prototypical open ocean speciation. So I agree that there are many reasons why you would not expect them to be the same. But that paper is widely cited as being representative of what phytoplankton look like in the ocean. And so I think it's important that someone stand up and say, actually, there's a lot of differences than what, I mean, they chose a manganese concentration, which was really high. It depressed their zinc and their cadmium quotas. So that, you're right. I'm not trying to hold their feet to the fire. I like but it's not, it's not the end-all be-all of what is out there in the ocean. That's my point. Yeah. Fair.
Any other questions?
Yeah, I have a question.
Yeah, Bob.
So I'm wondering if some of the picture of what you're talking about would change if you redistributed your sampling areas. I noticed that you had nothing from the equatorial West Pacific, which is where there's a lot of volcanic inputs. There are a lot of hydrothermal inputs into the surface waters. And so you kind of restricted yourself to, for the most part, the Atlantic and the Southern Ocean.
Yes.
So if you went to an area where there were these additional substantial inputs of metals, I'm wondering what the cell quotas would look like in those areas compared to where, or how you think they would look. And why do you stay away from those?
Well, I think it's unfair to say that I've stayed away from them. In addition to those stars, there's probably 10 more we could put up there. We've tried our best to collect samples from, wherever we can get them in some sense, where we think there are interesting local effects to look for. Would I expect to see differences? Well, based on the North Atlantic data, I would. It's interesting, the first place, after we started this by doing the Southern Ocean iron experiment, and we saw gradients after we had added iron. That didn't seem so surprising. But we then went, in this project with Nick and Stephen, to the Equatorial Pacific, where we went across the equator and along the equator, and we're hoping to see gradients and didn't see them. The cells were largely sort of the same moving across and along them, with some exceptions with tropical instability waves. So it has been more challenging, I will say, to see those gradients than I might have thought. In the FeCycle, actually one of my main goals in the FeCycle project was to look for temporal gradients. So we have this bloom, and we were interested in if the iron quotas are drawn down, for example, as the cells go from iron replete to iron limited before they stop growing and sink. And we didn't see strong gradients there. So actually what we see is that the cells, my interpretation is the cells have as much iron as they need, and we don't see the plasticity you see in the lab. Rather, once they move below their optimum concentration, they just can no longer compete, and you just don't see them anymore. You only see, I think the only cells that you see in the ocean are the ones that are at their prime, sort of competitive nature. So I was excited to see the North Atlantic gradients. I think it'd be great to go to an area, I mean the Western Pacific, where you have those strong inputs and to see if there are differences. If you've got some money, I'd be happy to do those for you. But no, it's really quite valid. I mean, I am following in a long tradition of taking a single data point and extrapolating it to an entire ocean basin. But we've added a lot more points since when I was here in 2003. So we will continue to try that. But yeah, there are definitely differences between those. All right, everyone's hungry.
Thank you very much.
Thank you.